I'm Brenda Wilson. There we go. Thank you very much for inviting me. Thank you for spending your evening listening to us and engaging with us. I just want to comment that you referred to us as experts several times, and I think one of the most exciting experiences for me is in the last year when I've served on this National Academies panel, none of us felt like experts. Gene drives is such a new field that we all felt like students. And it was really exciting to be in a room of people who are usually called experts, who are able to ask the simple questions and engage each other on an issue that is just emerging even as we were studying it. So I come to you as a student with a little bit of experience. I'm going to talk about the report that we released in June of this year called Gene Drives on the Horizon, Advancing Science, Navigating Uncertainty, and Aligning Research with Public Values. I'll just note that I'm part of the Genetic Engineering and Society Center at North Carolina State University, which is an interdisciplinary effort to look at issues of genetic engineering at the interface of society. And I was invited, show this slide just to show you how interdisciplinary the panel was at the National Academies. And this was really a testament to the priorities of the staff in this case. I'll talk a couple times about how different this report was to past National Academies reports. So gene drives, how many people have heard of them? Most have heard. I will talk a little bit about what they are and what they're used for, and then talk about some of the major findings from the study. I'm using a lot of the slides from our public presentation, so if you like them, that's great. If you don't blame me. But gene drives, what's interesting, and I should say this too, that gene drives are one example of what gene editing allows. And so Eric took us to the 40,000 foot level and thought that we might have to talk about the whole world tonight. I'm zeroing in on one particular technology, which is called gene drives. Gene drives have been proposed for these different areas: public health, conservation, agriculture, and basic research. For example, developing a mosquito that doesn't transmit malaria and having that take over a population. For conservation, using a gene drive to protect biodiversity by, for example, crashing a population of invasive rodents on an island. With agriculture, potentially changing pest populations or pest behaviors. And then for basic research in model organisms and others. So what are gene drives? They are systems of biased inheritance in which the ability of a genetic element to pass from a parent organism to its offspring through sexual reproduction is enhanced. So what that essentially means is that Mendel gets trumped. And I say that great hesitation in the United States. What it means is that the trait of a parent with a gene drive trait is very likely to be passed on, up to 100% of the time. So what that means is that through successive generations, you could essentially have the replacement of an entire breeding population with the trait that you introduced. What are the defining features of gene drives? What's really special about them is that they are designed to spread and they are designed to persist in the environment. And that is different from other GMOs we have seen contemplated or introduced thus far. As I said, there's two potential uses: population suppression, decrease the numbers of a particular population, or population replacement. So our report dealt with a lot of different issues around gene drives, and I'm really proud of the fact that this group of scientists were willing to think broadly about the issue of gene drives. National Academies studies are not always that way. And so we talked about the importance of thinking about human values. We talked about the scientific research that has been done and is underway. We recommended a phase testing approach, which I'll talk about. We had an expert in risk assessment who helped us think about what were the requirements for doing risk assessment with gene drives. We talked specifically about public engagement. To our knowledge, this is the first time that the National Academies has focused specifically on that issue. And of course, we talked about issues of governance. One of the major findings of our committee was that there is insufficient evidence available to support the environmental release of gene drives at this point. And yet, we also encouraged research to understand some of the uncertainties We feel it's a promising technology, but it's not ready for prime time. And that was something that was significant for our committee to say, given the pace of the research. We borrowed from the WHO's framework on introducing genetically modified mosquitoes to think about a phased testing approach. And we referred to this as a kind of precautionary approach in an effort to reclaim some of the discourse around what precautionary could mean. And so we envision, the print is pretty hard for you to see, but we envision various phases of research, and in each of those phases, there needs to be public engagement, there needs to be thoughts about governance. Each of those, moving from one phase to another, should have a go or a no-go decision. And so those phases include laboratory studies in phase 1, toward field-based research, staged environmental release, and then finally post-release surveillance, which is an important part of thinking about a technology that's meant to spread and persist. As I said, we talked specifically about public engagement. We envision public engagement as being complicated because there are not just the public out there. So publics are communities. For example, if you're introducing a gene drive modified organism into an environment, there is a community of people who live around that release site. There are stakeholders, which are broader publics, who may not have that kind of geographic proximity, but have interests and stake in this technology and its release. And then there are broader publics who may not be specifically connected to a particular instance of the technology, but whose values and opinions and perspectives matter in governing what could be a very powerful technology. We said a couple things that I think are quite notable. Public engagement cannot be an afterthought. That is the norm. That is the norm. We also said that the outcomes of engagement may be as crucial as the scientific outcomes to decisions about whether to release a gene drive modified organism into the environment. We got a group of scientists to agree to say that. That's a big deal. That consensus statement was something that I think is very, very significant about our report. We were recently attacked in the pages of Cell. I don't know if people saw that, but this kind of statement was upsetting to some people. So in terms of governance, we talked about many levels of governance around gene drive, from the personal responsibility of the scientist to professional guidelines, codes of ethics, things like that, through institutional reviews such as institutional biosafety committees, as well as what we think about in terms of regulatory structures that would impose specific guidelines or rules on gene drive research. What we did say was that at least, especially in the US context, the existing modes of governance don't consider gene drive's intentional spread and their potential irreversible effects on ecosystems. We also have a regulatory system where there's a lot of uncertainty about jurisdiction. There are insufficient structures for public engagement. We do have many public meetings. Those are not well attended by the public. It's not the best way to incorporate public views. Most of our regulations don't address the potential for misuse of these technologies, and there's a lack of policies for collaborating internationally. I know I'm going quickly, but I want to get onto our conversation too. One of the ways that jurisdiction is a problem is, for example, if a gene drive is inserted into a mouse, this would be to crash a mouse population as an invasive species on an island. The FDA considers genetically modified animals under its jurisdiction because they're considered new animal drugs. However, you could also think about that particular mouse as a rodenticide, which would fall under our Environmental Protection Agency regulations. It could also be a plant pest because mice are plant pests, and that's what's triggered the USDA's regulation of GMOs in the US. And so we have this new potential organism coming online, and we're not sure where it belongs in what we call our coordinated framework for the regulation of biotechnology. In addition, there are other US agencies that might need to be at the table, and that's not well facilitated by our current coordinated framework. For example, US Fish and Wildlife, the National Park Service, et cetera. The international perspective is really a tricky one, because these organisms, if they're released, they won't recognize political boundaries, and they're meant to spread. And so one of our case studies in our report talked about pigweed, which is an agricultural weed in the southeast United States that has become resistant to glyphosate. potentially you could use gene drive to take away the resistance in that population. A relative of pigweed is an agricultural crop, a food in Mexico. So what would it mean for us to introduce a gene drive in the United States context for something that's seen as a weed that could then spread to Mexico where it's considered a food? I'll just say that the coordination at the international level may be very important, and we don't have very good systems in place for this. So I just want to acknowledge that we had incredible staff at the National Academies. We did have members of the public who weighed in at various meetings. We went through a very, very rigorous peer review process, one that I've never seen before. And we had some fantastic community members who became close colleagues and friends. Our study was sponsored by the National Institutes of Health and the Foundation for NIH. I will just close by showing this slide, which is the reports of the National Academies are free for download. There's a report summary as well as a full report available at that website. Thank you very much.